Alright, so we will see the second subtopic in this chapter, which is the meiosis. So before we can start, we will see the overall of this subtopic. First, we will see what is meiosis is all about, the importance, and then some of the differences between meiosis and mitosis. And then we will have some terms that you must know. Okay, the meaning of homologous chromosome. Then we will see the stage of meiosis, and lastly we will see two processes briefly on how the gamete is produced. Okay, how the meiosis happen in the production of the gametes. Before we proceed with meiosis, okay, let us see about the importance of meiosis, okay, in human.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell.
The videos just now, okay, the short videos, we can see that, okay, meiosis is so important to create variation. Okay, the variation due to meiosis, okay, due to the half of the chromosome number produced during the formation of gamete. So we can get variation. Okay, so with this variation is very important. Yeah. Then you can see here another important is to maintain. Okay, to maintain the chromosome number from one generation to another generation. If the meiosis doesn't happen, alright, the number of chromosomes will increase from one generation to another generation. So the importance of meiosis is to make sure that the chromosome number is maintained. And if you see here, okay, to maintain the chromosome number is to form the gamete with half of the chromosome number, which is after the meiosis, okay, the gamete will have half of the chromosome number. After the gamete is formed, the gamete from the male and the gamete from the female will fuse to form the zygote. Okay, this is what you have learned before. Okay, just to refresh for me. Okay, so when the two haploid gamete fuse, okay, they will form one diploid zygote. So this is how the number of chromosomes is maintained. Okay, the new organism or the offspring that is produced from the fusion of the gametes will have the same number of chromosomes with the parent. Okay, just in case, okay, your gametes that we mentioned here, okay, the female one will be the ovum, okay, or ova, and the male one will be the sperm. Okay, and the place, the parts where the gamete is produced is called the gonad. The next term that we're going to see is homologous chromosome. So homologous chromosome is the chromosome that have about the same length with each other. So meaning to say, out of the 46 chromosomes that we have in our body, the human have, so the 23 of them is actually similar with another 23. That makes the homologous chromosome 23 pair in a human body. So each of these pair will come from one parent. Okay, from one parent. So one of it come from the mother, another half of it will come from the father. So we can see the homologous chromosome, they are similar in the size, the position of the centromere, the bending pattern, and in the staining procedure. So this one, the staining procedure means the one that is the karyotype. Okay, when we prepare the karyotype, you can see how it's being stained. So when it's stained, the color will be the same, the dark side, and then the bit or not dark. Okay, that one is in karyotype. Okay, then also they are similar in the type of genetic information they carry. And during the meiosis, these homologous, they will separate. Okay, later we will see how it separates in the videos. So the next one is the preparation of karyotype. So this is the karyotype that I meant. So we see the staining. If you use the multiple color, okay, the staining will be about the same for the homologous chromosome. And this is how we carry out our studies. Okay, next we will see the stages in meiosis. So different from mitosis, meiosis will have two main stages. Okay, the first stage is called the meiosis 1, and the second stage will be the meiosis 2, which we can see in the videos.
Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis 1 and meiosis 2. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells.
So we can see here the difference, the main difference between the mitosis and meiosis is the number of daughter cells. So the number of daughter cells in meiosis will be 4. After meiosis 1 and meiosis 2, we will get 4 daughter cells where the chromosome number is half, okay, the haploid daughter cell. As for in mitosis that you have seen before, okay, we will always get 2 daughter cells with the same number of chromosomes after the division. Okay, now we will see what's the difference in meiosis 1, or the overall pictures in meiosis 1 and meiosis 2. In meiosis 1, we will have all the 4 stages that you have learned in mitosis, which is the prophase, metaphase, anaphase, and telophase. But in meiosis 1, we will give a name for them by putting the number 1 there. So we will, instead of calling them prophase, we will name it prophase 1, metaphase 1, anaphase 1, and telophase 1. So end of meiosis 1, okay, end of meiosis 1, we will always get the cells, 2 cells, alright, 2 cells, just like, or 2 daughter cells, just like mitosis. But these 2 daughter cells will have haploid numbers of chromosome, which is the half of chromosome compared to the parent. And the same with mitosis, okay, prior to the meiosis, before meiosis, the cell will also undergo interphase, okay, interphase where they prepare to divide, okay. As you can see here in the pictures, okay, before the meiosis 1, okay, we have interphase. After meiosis 1, we have 1 cytokinesis. After that, meiosis 2, then cytokinesis again, okay. That means for one complete cell cycle for meiosis, they will have 2 times of cytokinesis, okay, then 1 interphase, then meiosis 1 and meiosis 2. Okay, it's something that you have to know lah. Okay, the whole cell cycle for meiosis. After meiosis 1, the 2 daughter cells, each of them undergo meiosis 2. Okay, under meiosis 2, the centromere will split. So from the splitting of the centromere, the sister chromatid will separate and become 2 daughter cells. This is how we get another 2 of daughter cell from each of the daughter cell in meiosis 1. So in total, we will have 4 haploid cells at the end of meiosis.
As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1 of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle.
From the video, we can roughly, or you can really see how the animation shows, on the prophase 1 of meiosis 1. Okay, now we will see after prophase 1, the metaphase 1, metaphase 1, and then until the first telophase end.
In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is two haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication.
Alright, what I need you to see here is, we have to compare it with meiosis and then mitosis. So in mitosis, the kinetochore is the spindle fiber, attached to kinetochore, the centromere. And then it separates the sister chromatid. But in meiosis, it separates the homologous chromosome. That is how the half of the chromosome is formed. Another key difference here, you will see that there is some parts which is stained blue. All the purple, you have some stained blue here. So this shows that crossing over happened during the metaphase stage. During metaphase, when they are aligned, some of the DNA or the information exchange, and we call this crossing over. What is crossing over? Crossing over, we can say, is a exchange of genetic materials. As you can see here in the videos, when parts of one of the member of the pair cross over to the homologous pair, we call this process crossing over. The point where they cross is called the chiasmata. The point where they cross is chiasmata. And also inside the video just now, they are saying about random assortment. So random assortment, as you can see here on the slide, on top of the slide, on the videos, we can see the chromosome align. So first possibility is all the blue on top, the yellow down. Then we can random assort one yellow on top, another one the blue top down. Then another possibility, is two yellow on top, the blue at the bottom. So this is called the random assortment. Then you can see there is part of it. If you have 23 pairs, you can think of how many random assortment that you can make. And that is how many possibility our genetics can be mixed. That means we have different variations here, a lot of millions of it. Then after that will be the metaphase 1, anaphase 1, and telophase 1. So they are almost the same in mitosis. So you just take note on the situation or the occurrence of inside. After the cytokinesis, we will have two haploid daughter cells. Then that's end for meiosis 1. Now we will see what happened in meiosis 2.
centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and cleavage furrows separate the two daughter cells into 4 haploid daughter cells.
So in conclusion, in meiosis 2, we have another 4 stages, which is the prophase 2, metaphase 2, anaphase 2, and telophase 2. And it ends with cytokinesis, where you have 4 haploid daughter cells.
The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent.
So as we said, the formation of the gametes, which is the sperm and the ovum. For the sperm, we call it spermatogenesis, and for ovum, it's called oogenesis. You can just read up the notes, okay? This is just a brief introduction to the 2 processes here. Okay, we will see this in later topic. So as we know that, the variation in the organism, is due to the crossing over and also the independent assortment during the formation of the gamete. So I hope you are clear, and then we will end this with some of the short videos that the end of the short videos, Thank you.
But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.